I'm the one of the clinical team members, and a lot of the work, all of the work so far has been preclinical and on the basic science side. So, they can speak to it way more than I can. However, the encoded concept, I think, makes a ton of sense from a clinical standpoint. It has a lot of advantages, and the gist of it is that they're taking a well-established AAV gene delivery platform, and the fact that it's well-established helps us already kind of gain certainty about safety and tolerability because it's been used in humans before, and delivering a mechanism to increase SCN1A expression. And SCN1A expression is the way that I say it, rather than increasing SCN1A copies, because they don't actually deliver an SCN1A copy. They deliver a kind of a custom regulatory element which will select for the interneurons, the GABAergic interneurons, which are inhibitory. That means that you won't have off-site effects in other cells. Then you have additional selectivity in terms of using an engineered transcription factor, which will preferentially express the SCN1A protein by creating a protein which upregulates SCN1A expression. So unlike kind of the traditional model of, or the default kind of mental picture that people have of taking an extra copy of SCN1A and putting it into the nucleus somewhere, the approach here is actually putting something into the nucleus which, when translated, will increase transcription of SCN1A. So it's a very regulation-centric model rather than a copy number model. And so I think that approach brings down the size of the payload that's needed to induce the effect. It makes it possible to deliver in this well-characterized, relatively safe virus, and makes it really exciting for patients. So I prefer the term SCN1A related epilepsy to Dravet because Dravet includes other genes also. In some cases, 90% of Dravet syndrome is caused by SCN1A mutations, and this is only for SCN1A. So it's really SCN1A related epilepsy. The other challenge that comes up with that terminology is that before the natural history of the patient has really declared itself, you don't know whether they will have Dravet or whether they will have a severity that's somewhere else on the GEFS plus spectrum. And so SCN1A related epilepsy really captures all of it. And anybody who has that, even if they're young enough that Dravet is uncertain, yes or no, it's not important. As long as SCN1A is the cause, then this is an appropriate therapy. The thing that's really important to understand about the therapy is that because it's genetic, it has the ability to address parts of cognitive function that no other treatment that's available today can even hope to address. That's the direct effect of gene dysfunction on learning and cognition. We have treatments that can help learning and cognition because seizures bring it down, and so we'll stop the seizures and we'll allow that to float back up and hopefully improve. We have medications that are less sedating and have less intoxication effects on cognition and behavior, but this is the only thing that can touch the direct genetic effect on cognition and behavior that other conventional anticonvulsant medications can't.